All right, so let's begin the second half of chapter 22. We're going to talk about the small intestine, the large intestine, the pancreas, liver, gallbladder, and then we're going to put it all together. But first, let's talk about the small intestine, sometimes called the small bowel. Ironically, is actually the longest portion of the alimentary canal, uh, at about 20 feet long. In a living person, of course, this is much shorter due to smooth muscle tone. And there are four main processes that occur in the small intestine. There's secretion, digestion, absorption, and propulsion. Now, the cells of the small intestines, often called enterocytes, they're going to produce multiple digestive enzymes, multiple hormones, as well as lots of mucus. And these enzymes, along with those released by the pancreas, are going to be responsible for the bulk of the chemical digestion. And in fact, no more nutrient digestion actually takes place once food leaves the small intestine. Um, Although technically bacteria in the large intestine do break down certain nutrients, they're not really um, digested by any enzymes that are created. After these nutrients are digested chemically, they're going to be absorbed across the enterocytes into either the blood or the lymph, along with water. It's going to be absorbed with water, vitamins, several other substances. And then, like the stomach, the small intestine also mixes and propels its contents along its length and into the large intestine. And the small intestine really has three divisions. There's the duodenum, the jejunum, and the ileum. And they're distinguishable by several very slight histological differences. The first one is the duodenum, and this is that first segment of the small intestine that starts right up against the pylorus of the stomach. And it is the shortest of all three divisions, only about 10 inches long. This duodenum really arches itself into a C shape as it curves around the pancreas, and only the proximal portion of the duodenum, the one closest to the stomach, is within the peritoneum. The remainder of it sits posterior to the peritoneal cavity and is thus considered retroperitoneal. Internally, the duodenum is going to house the major duodenal papilla, and these are secretions from the gallbladder and the pancreas. Uh, these are where, secretions from the gallbladder and the pancreas are going to enter the small intestine. And there are several specialized glands called Brunner's glands or duodenal glands that work to produce an alkaline mucus that basically protects the duodenum from all of that acid that has come from the stomach. You also have the jejunum, and the jejunum is the middle portion of the small intestine. It's about 7.5 feet long, and this is going to begin at what is called the duodenojejunal flexure. And this sits within the peritoneal cavity. And it is the most active site for chemical digestion as well as absorption. And then the last section you have is the ileum, and this is the small intestine's final segment, also considered intraperitoneal, and it's about 10.8 feet long, almost 11 feet long, and it terminates at the portion of the large intestine called the cecum. There is a juncture there called the ileocecal valve that is a ring of smooth muscle. So that is a sphincter. That is the ileocecal sphincter. And this controls the movement of material from the ileum into the cecum. This sphincter is also going to prevent materials in the large intestine from ever backflowing into the ileum. And this is very important because the large intestine actually houses a great number of bacteria that, if it entered the ileum, would cause a significant illness. And here you have that image. You see, let me pick up this color. You see that pyloric sphincter joining up with the duodenum. And as we said, the duodenum is very short, heading around that duodenal flexure. or rather heading, curving around the pancreas may be a better way to say that. Um, After that, you do have the major duodenal papilla, which is going to secrete those pancreatic and gallbladder secretions. After that, you have the middle section, the jejunum, here you see in green, and it has the large amount of chemical digestion and absorption. This is where the majority of that nutrient absorption actually takes place. And if you notice this web-like material that connects all of it, that is the mesentery, and we've talked about this already. The mesentery is just another part of that peritoneum, that visceral peritoneum that connects everything and helps hold its shape. But eventually, the jejunum joins up with the ileum, and this is the last section of the small intestine, which flows all the way down to the large intestine and joins up here at the ileocecal valve. And it's important, that ileocecal valve, because it prevents backflow of dangerous bacteria into the small intestine. And then this little thing you see here, actually, we'll talk about it again later, but that is actually your appendix. That is what the appendix looks like. Now, which site of the small intestine is the site of the majority of absorption? It's the jejunum. And it is important to remember that the small intestine remember those same 4 layers of tissue, the serosa, the muscularis externa, the submucosa, and the mucosa. But as always, the form of the small intestine is going to have specializations that allow it to better perform its functions. Now, the internal surface of the small intestine is actually going to contain 3 progressively smaller types of folds. So each fold is going to be smaller than the last. And most of the absorption that occurs inside of the small intestine, and so these folds are able to increase that surface area available about 400 to 600 times what it would be if it was just smooth. Now, the largest types of folds, which are visible to the naked eye as essentially ridges in the walls, these are called circular folds, or plicae circulares. And these circular folds involve both the mucosa and the submucosa of the small intestine. And they do not only increase surface area, but they also work as speed bumps to slow down chyme through the small intestine. And then that gives nutrients more time to be digested. And the small intestine cells, which are, remember, those enterocytes, it gives them more time to absorb those nutrients. And here you can see the circular folds, which again are visible to the naked eye as ridges. So that's the ridges you see inside this small intestine here. Now, you also have 2 smaller types of folds not visible to the naked eye. The mucosa is actually going to fold itself into projections called villi. And these villi consist of a layer of enterocytes and the occasional goblet cells that secrete mucus, all of these surrounding a central core of blood capillaries and lymphatic vessel called a lacteal. Now, in between each villus, the mucosa is going to indent to form what is called an intestinal crypt. And each of these house glands that also contain hormone-secreting DNES cells. And here you can see the DNES cells down here in the crypt. Well, let's see in between each villus, you see this crypt, and you see these hormone-secreting DNES cells. All of these around this big projection called a villus. Multiple are called villi. And again, you see it's simple columnar epithelial tissue with mucus-secreting goblet cells. And this lymph tissue, this lymph duct here is the lacteal, and these are blood capillaries you see here with venules and arterioles here, feeding and draining each villus individually. Now, the smallest folds, remember I said there are two types of small folds. These are called the microvilli, and they're actually found on the plasma membrane of the enterocytes. Each one is going to have as many as 3,000 microvilli, which give the cell the appearance of a sort of bristled brush or a brush border. And associated with this brush border are numerous digestive enzymes that are produced and secreted by these enterocytes, like sucrase, maltase, lactase. All of these work to catalyze reactions that break down sugars, as well as several proteins. And then they also catalyze reactions um, that work to break down several peptides, several proteins. And so we can disregard this and only look here at the enterocytes. And you see this sort of bristled brush border here made up of the microvilli. And remember, there are about 3,000 of these on each enterocyte, and each one of these quote-unquote simple columnar epithelial cells is coated with this brush border. All of this working to increase the surface area, allowing for more easily digestible nutrients. Now, it turns out the speed at which the small intestine contracts and pushes food through actually differs during fasting and eating. During a fasting period, the small intestine will exhibit slow, rhythmic contractions along its length in a pattern called the migrating motor complex. These contractions work to clear any remaining material from the small intestine, including leftover food and any secretions that are there. And this takes about 2 hours to push any digesting food from the duodenum to the ileocecal valve. And all this movement is going to be controlled by both the enteric nervous system as well as a hormone called motilin, which is going to be produced by cells in the duodenal mucosa. And the small intestine also undergoes two different types of movement while eating. There is, as with other regions of the alimentary canal, there's peristalsis, and this is going to be accomplished by alternating contractions of the longitudinal and circular muscles of the muscularis externa. And its primary function is to propel chyme toward the ileum and ultimately through that ileocecal valve. However, there is another process called segmentation, also known as intestinal churning. And this involves contractions of only the circular layer of smooth muscle, which produces a sort of squeezing motion. And this segmentation is really a form of mechanical digestion as well as mixing, and it works to mix that chyme with intestinal and pancreatic enzymes as well as bile from the gallbladder. And the vagus nerve really appears to regulate both peristalsis and segmentation. And here you can see peristalsis, just this rhythmic propulsion. All of the material is traveling in one direction. But then you have just the smooth muscle inside contracting, and it causes, yes, this chyme to sort of churn and mix and just make sure that all of the food material is bathed in this pancreatic juice and this bile from the gallbladder to really just make sure any chemical digestion is done as efficiently as possible. Now, this table has all of the hormones that are going to be involved in both secretion and motility of the digestive organs. And every single one of these is important. And so you guys are going to need to know what each hormone is, what its stimulus is, as well as what effects that hormone has. So I'll give you a second just to pause this and be able to go over this on your own and memorize this. Now let's just review just a little bit. Which of these is the first process to occur in the digestive system? It's mastication. That chewing of the food is really the first thing that has to happen. Technically, if you really want to get super duper in the weeds about it, secretion would be, as long as you're only talking about the salivary glands. But that's super in the weeds. The actual what we're going to look for here is mastication as the first process. Now, which of the following catalyzes carbohydrates in the mouth as well as the stomach? It's amylase. Amylase really breaks down those starches and those carbohydrates, and it is secreted by saliva. Now, what is it that chief cells release? Chief cells work to release pepsinogen. And remember, that's that inactive form of pepsin that is activated by the acid that is released by parietal cells. So chief cells release the pepsinogen, acid released by parietal cells activates it, and then it becomes a sort of peptase that can break down peptides. Which segment of the small intestine receives chyme from the stomach? So this would be the first section of the small intestine, right? Because it has to butt up against the stomach. It's the duodenum. The duodenum is what's going to receive all the chyme from the stomach. It's also going to work to neutralize that highly acidic chyme with its alkaline mucus. Now we can talk about the large intestine, otherwise referred to as the large bowel. And this is going to run along the border of the abdominal cavity, working to sort of snake around the small intestine as several other abdominal organs, sort of like a frame. At about 5 feet long, it is actually named the large intestine because it has a larger diameter than the small intestine, despite being much shorter. Now, the large intestine is going to receive any material from the small intestine that was not digested or absorbed. And so this, you might think, the small intestine does all the work, and then the large intestine is simply a passageway for leftovers called feces. But that's not really the case. The large intestine is really mostly active in absorbing water and electrolytes. Think of all of the water and all of the secretions that have been pumped into this chyme. If we just passed that through, we would be losing a great amount of water. And so the large intestine works to reclaim a lot of that. And when this absorption, this water absorption or electrolyte absorption is interrupted, such as with bacterial or viral gut infections, a person can really easily become dehydrated, as well as electrolyte or acid base disturbed. And so this is the large intestine here. We see down here, we see the ileocecal valve, and then down here you see that vermiform appendix. But the ileocecal valve dumps from the ileum here, into the cecum here. And so this cecum is just the first section of the large intestine. And then we go up the ascending colon, around the hepatic flexure, across the transverse section, down the splenic flexure, down the descending colon, around the sigmoid colon, out the rectum, and then finally that waste is eliminated through the voluntary contractions of the external anal sphincter and exits the body. Now, the cecum is going to be that first section. The first section of 3, technically, the cecum, the colon, and the rectum. Now, the first portion is the cecum. This is called the blind pouch that is intraperitoneal and located in the right lower quadrant. And this does house that smaller blind-ended pouch called the vermiform appendix. Vermiform just means worm-like. And this is simply just often shortened to appendix. And for a long time, the appendix was considered a vestigial organ, or one left over from evolution that we really didn't have any function. But now it's shown to house multiple lymphatic nodules, and it really works to play a role in the immune system. And despite its small size, it does have big problems if it becomes inflamed. We'll talk about that when we get to appendicitis. And so the cecum is just going to be this first area here, this sort of blind pouch where the ileocecal valve dumps into. Now the next and the longest portion of the intestine is called the colon, and this is divided into 4 subsections. You've got the ascending colon, the transverse colon, the descending colon, and the sigmoid colon. The ascending colon is going to be retroperitoneal and travels superiorly along the right side of the abdomen up to the liver. And when it hits the liver, it's going to make a sharp left-hand turn at what is called the hepatic flexure, also called the right colic flexure. After the hepatic flexure, the ascending colon becomes intraperitoneal transverse colon. So it's still the same tube, it's just now it is traveling transversely across the superior abdominal cavity. Once it hits the spleen, it's going to take a sharp turn inferiorly at what is called the splenic flexure. Are you getting this? The splenic, the spleen, the hepatic, the liver. So at the liver, it takes a hard left turn, and then at the splenic flexure, at the spleen, it's going to take a sharp turn down. And once it begins turning down and traveling downward, this is now the retroperitoneal descending colon passing along the left side of the abdominal cavity. Eventually, this descending colon is going to become an S-shaped sigmoid colon. It's bending and it's becoming a little bit narrower until it becomes the rectum. And this is the portion of the large intestine that runs anterior to the sacrum and is retroperitoneal. The walls of this rectum feature several horizontal folds called rectal valves, and these work to allow the flatus, the gas, without risking the simultaneous passage of feces. In simple terms, these rectal valves are what allow farts to go through without pooping your pants. Got a little carried away and forgot to hit next on the slideshow. So this says a lot of the same stuff. The rectum is going to end at the anal canal, which is the distal portion of the large intestine. And I want you to remember from the muscular system chapter, the anal canal passes through what is called the levator ani muscle in the floor of the pelvic cavity. And the walls of the anal canal are going to feature longitudinal grooves called anal columns. And in between each column is what is called the anal sinuses. These secrete mucus when feces pass through the anal canal during defecation. And as the terminal portion of the anal canal has 2 sphincters, the first is called the involuntary internal anal sphincter, and this is simply a thickened layer of the circular muscle from the muscularis externa, and it is supplied by parasympathetic motor neurons. The other one is the voluntary external anal sphincter, which is composed of skeletal muscle. And as this sphincter is voluntary, it is innervated by somatic motor neurons controlled by the cerebral cortex. So here we can see some of those structures I was talking about. The rectal valves. Here you can see some of these anal canals being carved out, and in between each one you have those anal columns. In between each anal column you have the anal sinuses. The anal canal is this actual lumen of the alimentary canal here. Outside here you have this external anal sphincter, not to be confused with the internal anal sphincter, which is involuntary, all of which sort of pushes through, pierces the levator ani muscle. Now the splenic flexure, this is a test yourself question. The splenic flexure is found between which 2 structures of the large intestine? So the blank curves at the spleen and becomes the blank. The transverse is going to turn downward and become the descending colon. And then which sphincter of the anus is actually voluntary, controlled by the cerebral cortex? It's the external anal sphincter that is controlled by the brain voluntarily. Now, before I get too off topic, I want to talk about appendicitis. And it's really important because the small size of the appendix and the fact that it is a blind-ended, which means it just has one opening and no other exit, this means that it can actually occasionally become blocked. Generally, this block is by fecal matter, and then bacteria in the feces are going to multiply inside the appendix and cause an infection. And that infection results in inflammation, and this condition is called appendicitis. Telltale signs of appendicitis are lower right quadrant abdominal pain, rebound tenderness similar to that experienced with peritonitis, nausea, and vomiting. And appendicitis really does require immediate treatment. Typically, it's an appendectomy, the surgical removal of the appendix. If it is not treated, the appendix could continue to swell and could very easily rupture, not only causing internal bleeding, but that bacteria-filled fecal matter is going to spill into the peritoneal cavity, leading to a further infection of peritonitis. And these two conditions combined can easily be fatal if they're not properly managed. So appendicitis is a very serious condition. Right lower quadrant abdominal pain is that telltale sign. Now, histology of the large intestine. Remember, we talked about all of the layers are pretty much the same, but they're going to be specialized now. So the mucosa of the large intestine is going to lack villi, and its cells lack microvilli. So where the small intestine had the projections, the finger-like projections, and then the brush border, these now are not here. And this really reflects the lack of nutrient absorption, as that is not the large intestine's primary function. And much like the rest of the alimentary canal, this mucosa is instead rich with goblet cells that secrete a protective and lubricating mucus. And the muscularis externa of the large intestine is going to be special in that its longitudinal layer is really not continuous throughout most of its length. Instead, this layer has been, over time, has been condensed into 3 bands of muscle called the tinea coli, and their constant tension is what bunches the colon into small pockets called haustra. A single pocket would be called a haustrum. Now, the serosa, or the visceral peritoneum, of this large intestine is going to contain several fat-filled pouches called the epiploic appendages. These actually, no one knows what they do, but they're just fat-filled little pouches that hang off near the tinea coli, and we should see those in just a second. So here you can see these epiploic appendages here, and especially you can see the tinea coli here, these 3 bands of longitudinal muscle that have been condensed. They've sort of like this side has been brought around and condensed here, and that side has been brought around and condensed here. All of this working to bunch these up in these haustrum. Each of these little pouches along the large intestine. I just wanted to make sure you guys were able to see this as we move along. Now, it turns out the large intestine hosts a truly tremendous number of bacteria. About 10 times more bacteria exist inside the large intestine than there are cells in the human body. You, the person listening to this, are 10 times more bacterial cells than you are human cells. And this bacteria can actually make up about as much as 60% of the weight of the dry mass of fecal material. These bacteria are called normal flora or gut flora, and there are about 500 different bacterial species that typically coexist with humans in a sort of symbiotic, mutually beneficial relationship. The humans are able to provide these bacteria with an environment they need to survive, and the bacteria in turn manage to perform a number of useful functions, like they produce vitamins, like vitamin K, which is necessary for blood clotting. They're able to metabolize undigested materials, like soluble fibers that the small intestine is unable to digest, converting them then into fatty acids and other compounds that the body can absorb and use. This is also going to aid certain vitamin and electrolyte absorption. However, an unfortunate byproduct of this is that gas that is created and released as flatus, farts. It can actually, these bacteria that exist, our gut flora, can actually prevent the growth of any pathogenic or disease-causing microorganisms by outcompeting them for nutrients and providing chemicals, or producing chemicals rather, that are actually biocidal for certain harmful bacterial species. In addition, during infancy, these normal flora produce an immune tolerance to their own antigens. So at the same time as an infant, these bacteria stimulate the development of mucosa-associated lymphatic tissue, MALT, and the production of any antibodies to pathogens. And so this actually helps to create a more favorable environment for the normal flora while protecting the host from pathogenic bacteria. And before I go too far off, antibiotic therapy, like long-term antibiotic therapy, especially those taken by mouth, can really disrupt the gut flora by killing off large portions of it, producing large numbers of problems, especially infection with antibiotic-resistant pathogens like Clostridium difficile. If anybody's ever worked as a CNA and they've had a C. diff patient, you know exactly what I'm referring to. This is a diagnosis that you can smell from across the room in the elevator. And I do want you to note that this situation can also occur in individuals who unintentionally consume antibiotics by eating antibiotic-fed meat. So those antibiotics stay within the muscle and then can easily be passed to those who eat that meat. And the large intestine now has two functional segments. We're going to talk about the proximal large intestine, consisting of the ascending and transverse colon, and the distal large intestine, consisting of the descending sigmoid colon and the rectum, as well as the anal canal. The proximal large intestine is going to be the primary site of that water and electrolyte reabsorption, as well as bacterial activity. There are two types of motility in this proximal region. The first is going to be a type of segmentation or churning, similar to what we saw in the small intestine, where the circular muscle of each haustrum contracts repeatedly, swirling the material around inside of it, aiding in water and electrolyte absorption. Contractions like that are going to be controlled by local enteric neurons and triggered by any stretch that occurs in the membranes. Now, aside from segmentation, there is also a propulsive motion called mass movement or mass peristalsis. Now, during a mass movement, multiple haustra are going to undergo peristalsis at once, which propels their contents toward the distal large intestine. Mass movements really just occur 3 to 4 times a day and really just appear to be triggered by food consumption, which initiates several reflexes controlled by the enteric nervous system. Now, there is a small amount of absorption that takes place in the distal large intestine, and it's mostly of water. But the distal large intestine's main role is to store fecal material until it is ready to be expelled during defecation. And so, for this reason, the distal region of the large intestine is much less motile than the distal region is less motile than the proximal region of the large intestine. Gosh. And when the mass movements then force fecal material into this normally empty rectum, it's going to initiate the parasympathetic mediated defecation reflex, in which the walls of the rectum housing stretch receptors are going to be activated by the presence of fecal material stretching out the walls. Sensory neurons are going to transmit those impulses to the spinal cord. Typically after that, the parasympathetic neurons are going to cause smooth muscle inside the sigmoid colon and the rectum to contract, and the internal anal sphincter is going to relax. And in an infant or a young child, this relaxation of the internal anal sphincter is going to result in the release of fecal material. In older children and in adults, the defecation reflex ends here because there is conscious contraction of the external anal sphincter. You're able to judge social cues, whether or not this is an appropriate time to defecate. The reflex is going to be initiated again with the next mass movements, and steps 1 and 2, the stretch receptors transmitting sensation and the parasympathetic neurons causing smooth muscle to relax, are going to be repeated. But now let's say you're on the toilet, defecation is proper, you're ready to defecate. So now impulses from the cerebral cortex are going to trigger the relaxation, the voluntary relaxation of the external anal sphincter, also contracting the levator ani muscle, elevating the anus. All three of these movements combined allow feces to pass. And do note that this activity is assisted by the contraction of abdominal muscles against a closed glottis. A procedure called the Valsalva maneuver can also increase intra-abdominal pressure. And you may have never heard it called that, but the Valsalva maneuver is essentially that strain you do when you close off your air passages and still it feels like you're trying to press air out of them. It's that that grunt sort of pressure. If you hold your nose closed and your mouth is still closed, but you're trying to force air out, you're actually going to increase abdominal pressure while you do that. And so this is that reflex that we're talking about. And the first thing we're seeing is that stretch receptors in the anus are going to receive this signal of rectal distension, and they're going to transmit those signals to the spinal cord and eventually actually get processed there. Parasympathetic neurons are then going to cause that smooth muscle in the sigmoid colon to contract, and the internal anal sphincter is going to relax. So this is going to push the fecal material toward the external anal sphincter. And as I said, in an adult, if this is not an appropriate time to defecate, the external anal sphincter is going to remain closed. If the person is sitting on the toilet, they're going to consciously trigger relaxation of the external anal sphincter and the levator ani muscles, allowing that fecal material to pass through and exit the body. But here you can just see those neural pathways being traced out. All right, so what is the normal, naturally occurring bacteria of the large intestine? What is that called? You said normal fauna. You're wrong. That's natural flora. The difference there is animals versus plants. When this was all named, bacteria was considered plant-like. So that's where it gets that name, normal flora. So anything that is going to change the motility, the amount of motion of the large intestine, is really going to affect the amount of water that is present in the fecal material. When the motion of the large intestines is increased, it doesn't have enough time to really absorb as much water from fecal material. And so this is going to produce a watery feces. This is a condition known as diarrhea. There are a number of factors that can increase motility, like irritation of the colon due to bacterial or viral infections. There's drugs that stimulate the parasympathetic nervous system. But in the opposite direction, when motility decreases, the large intestine can actually absorb too much water, and the fecal material can become very hard. And this is a condition called constipation. And the large intestine's motility is typically slowed by some drugs like opiate narcotics and anticholinergic drugs. So the figure you guys are about to see is really just a summary of the structural and functional properties of the large intestine as well as the various other organs of the alimentary canal. So you see the oral cavity, you see the structures within it and its functional roles, the pharynx, the esophagus, the stomach, then the small intestine, all 3 portions, and the large intestine, all 4 portions. Sorry, 5 portions. Cecum, colon, rectum. No, 3 portions, 5 subsections. Cecum, ascending colon, transverse colon, descending colon, sigmoid colon, and rectum. So now we're going to talk about some of the accessory organs that we've talked about just a little bit, like the pancreas, liver, and gallbladder. And these are going to be just like the salivary glands. These are exocrine organs. I'm sorry, these are exocrine glands that work to secrete a product through a duct to the outside of the body. And you might find this strange, considering the accessory organs secrete their products into the alimentary canal. And it logically appears that the alimentary canal is inside the body. However, because the alimentary canal is open on both sides to the environment, this is considered outside the body. Think of it like a paper towel roll. The outer surface is akin to the body's skin, and the inner surface is like the alimentary canal. Both surfaces in contact with the outside environment, and so they are thus considered external. So we're going to work through these in order from pancreas to liver to gallbladder, and we're going to look at neural and hormonal signals regulating them and any secretory products that come out of each organ, as well as the roles therein. So I want you guys to remember that the pancreas is going to be that endocrine gland that also has exocrine functions. The endocrine secretions are going to be insulin and glucagon released into the blood, affecting most cells in the body. The exocrine secretions are really just enzymes secreted by those acinar cells. And these enzymes are going to be released into ducts of the pancreas that empty then into the alimentary canal, assisting with digestion. We're going to look here at the structure of the pancreas and the acinar cells, as well as some of their secretions here. Now, when I show you this photo of the pancreas, you're going to notice that it has a wide head that contacts the duodenum. It has a middle width body, and then it has a much thinner tail tapering off toward the spleen. And then running down the middle of it, you have the main pancreatic duct, which receives the secretions from those acinar cells, funneling them into the duodenum. And near that duodenum is where that duct merges with the duct from the liver and the gallbladder, so all of them sort of empty in together. And acinar cells themselves are really just simple cuboidal epithelial tissue, and they're found in clusters called acini, and these acini surround a small duct into which they then secrete their products. Most of these duct cells also secrete products, but the main thing here is that these small ducts merge and drain into the main pancreatic duct, while some of them secrete into a smaller accessory pancreatic duct. And so here you can see the pancreas itself. You see this wide, you see this wide head, a middling, body, and then a thin tail. And you also see the common bile duct coming down from the gallbladder, meeting up with this main pancreatic duct right here, and then both of them end up in the major duodenal papilla, emptying their secretions there to assist with digestion and break down any nutrients that we can absorb. And then over on this side, you see how the Ossinar cells are sort of clustered together, and they all sort of secrete down into a duct, which would be this lumen area here, and that duct will then just carry those down into that main pancreatic duct itself. So you got smaller ducts coming out of the pancreatic ossini, and then they all sort of flow in the same direction all the way into the duodenum. Now, collectively, anything produced by the ossinar and the duct cells, these are going to be known as pancreatic juice. And this is water, as well as several enzymes and proteins, as well as bicarbonate ions, which remember, as I told you, this chyme coming out of the stomach is very, very acidic. And so these bicarbonate ions help to neutralize that acid, and that protects the duodenum from any damage. These digestive enzymes that are secreted by ossinar cells, they are crucial in chemical digestion, and they work to catalyze the reactions of carbohydrate, protein, lipid, and nucleic acid digestion. And like gastric secretion, pancreatic secretion really occurs at a flat basal rate between meals. When feeding activity occurs, pancreatic secretion is going to rise due to parasympathetic and hormonal stimulation, typically due to a hormone produced called cholecystokinin, or CCK. And this is produced by duodenal DNES cells in response to the presence of lipids and partially digested proteins in the duodenum. So the CCK is going to act on these ossinar cells, triggering them to secrete more digestive enzymes and other proteins. There's another stimulatory hormone called secretin, which is released by duodenal cells, and this is in response to acid and lipids in the duodenum. And it really primarily triggers duct cells to secrete some of those bicarbonate ions. So if acid is in the duodenum, it's going to trigger the secretin production to try and balance out the acid-base balance. And here you can see some of that. CCK being released into the blood, traveling to the ossinar cells, and then they secrete their enzymes. You have some secretin cells here being released, which are going to bind to the duct cells here, and they work to secrete those bicarbonate ions. So at the same time, you've got CCK producing enzymes, secretin producing bicarbonate, all of it working together to protect the duodenum and break down those nutrients. Now, where is cholecystokinin produced again? CCK is produced where? CCK is produced in the duodenum, and then it travels to the pancreas. Don't forget that. Produced in the duodenum, travels to the pancreas. Now let's look at the liver and the gallbladder. And the liver is going to be this pyramid-shaped organ. It is actually the largest organ in the body aside from the skin, and it is nestled in the upper right quadrant against the inferior surface of the diaphragm muscle. So just under the diaphragm, you have the liver. On the posterior side of the liver, the back of the liver, you have a small sac called the gallbladder. Typically, this gallbladder is very, very green and very easy to identify. Uh, the liver and the gallbladder have a very close anatomical and functional relationship, so we're really going to talk about these together. We're going to talk about them and as far as anatomy and histology, their basic functions, and their main digestive secretion called bile. But first, the liver is really just going to be wrapped up in a thin connective tissue capsule, and most of it's going to be covered by that visceral peritoneum. And it has four quadrants, four lobes. You have the large right and left lobes located on the right and left sides of the liver. Then you have the smaller caudate and quadrate lobes located on the posterior side of the liver's right lobe. Now, the right and left lobes are separated by a fold of the visceral peritoneum called the falciform ligament. And this also works to anchor the liver to the anterior abdominal wall. On its inferior surface, you're going to find something called the round ligament, which is actually the remnant of an umbilical vein that was present when the person was a fetus. And here you can see those structures I was talking about. We have the falciform ligament joining the right and left hemispheres, the right and left lobes. And so you see the right lobe, you see the left lobe. On the back down here, you see the gallbladder, and you have this round ligament, the remnants of that umbilical vein. On the back of the liver, you find an indentation called the porta hepatis, and numerous blood vessels enter and exit the liver here, including the hepatic artery, the hepatic portal vein, and in addition to these blood vessels, you're going to find several nerves, lymphatic vessels, and the common hepatic duct entering and exiting the porta hepatis. And note that in the hepatic veins, which drain blood from the sinusoids, they empty into the inferior vena cava on the liver's superior side. So here you can see some of those. So remember that blood is flowing into the liver from the rest of the body through this structure here, the hepatic portal vein. And so yeah, the liver is really filtering out all of the blood here, and then it's traveling out through the hepatic vein into the inferior vena cava here. All of this is drainage. Feeding the artery, feeding the liver here with oxygen-rich blood, you have the hepatic artery. You also have several nerves. It looks like none of them are actually labeled here. But here you have the quadrate lobe, and here above you have the caudate lobe. In addition, you have a much clearer view of the gallbladder, as well as the common bile duct. Now let's look at the histology of the liver. So if we're talking about the cells of the liver, the basic unit of a liver is going to be called a liver lobule. And these liver lobules are going to be separated by one another via septa that branch in from the connective tissue capsule of the liver. And a liver lobule is really just going to be composed of flattened plates of cells, remember those hepatocytes, and these are going to be arranged in the shape of a hexagon and stacked on top of each other. In the center of the lobule, you're going to find a central vein. And then at each of the 6 corners, you're going to find 3 structures collectively referred to as a portal triad. You're going to find a branch of the hepatic artery called the hepatic arteriole. You're going to find a branch of the portal vein called a portal venule. And you're going to find a small bile duct that carries bile. And the hepatic arteriole and the portal venule both drain into those large leaky capillaries, and the hepatic sinusoids that pass through or between rows of hepatocytes. And blood is going to flow slowly through these sinusoids as materials, as gases and nutrients are exchanged between the blood and the hepatocytes. Eventually, they're going to drain into the central vein. These central veins are going to merge and drain into the hepatic veins, which then in turn feed into that inferior vena cava. Do notice that bile is going to flow through the liver lobule in the opposite direction from hepatocytes into tiny ducts called bile canaliculi. And these are eventually going to drain into a common bile duct. And so here you can see a hepatocyte both in an image and in a micrograph. In a micrograph, it's pretty hard to see anything except that central vein. That central vein is really always easy to see, and then you do see that it is shaped sort of hexagonally. This edge here is a little harder to see. But you do see 1, 2, 3, 4, 5, 6 edges, as long as you use your imagination and say that that's an edge. But then again, you do see, okay, so the blood is going to flow in via the hepatic arterioles and portal venules. It's going to flow across the hepatocytes and then drain into the central vein. All the while, bile is actually going to travel the opposite direction down these bile ducts into a common bile duct eventually. So these bile canaliculi are going to eventually empty into one of these bile ducts, which empty into the common bile duct. So as, let's see, what's a good color that'll show up here? So as blood flows this way, bile's actually going to flow that way. It's just a really interesting thing to note. Now, which of these is not a component of one of those portal triads that I just mentioned? Remember, there are 3 things in there, and one of these is not one of them. It's the pancreatic arteriole. The pancreas has nothing to do with the portal triad because the portal triad is in the liver. Try not to get those confused. So now let's look at the functions of the liver. And the liver is really has a really great diverse set of functions. Much like the pancreas, it releases both endocrine and exocrine secretions, but it also converts harmful chemicals into non-toxic substances that can then be eliminated via waste. And so we're going to look at these functions over the next 2 sections. There's going to be bile production, which is crucial, as well as several other functions that are just going to be clumped together. But let's look at bile production first. And that is its main digestive function. And this bile is really a liquid that contains multiple components, including water, electrolytes, and several organic compounds. And bile does serve 2 critical functions. It is required for the digestion and absorption of lipids, so fats, and it is the mechanism by which the liver excretes wastes and other substances that the kidneys cannot excrete. And so one of these main organic compounds is called bile salts. These are going to be created from cholesterol, and they're amphiphilic, meaning that they have both polar and nonpolar parts. This allows them to interact with both lipids and the watery environment of the small intestine. So when this bile is released into the duodenum, those salts coat the lipids and physically break them apart into smaller pieces, and this is called emulsification. And this emulsification is the mechanical digestion of these lipids, and it is necessary for the chemical digestion as well, which we're going to look at in a little bit. Now, it turns out, bile also contains a varying amount of other materials that come from the liver, like cholesterol, waste products, toxins like heavy metals, and many of these are not going to be resorbed, and they're just going to pass through into the feces. One of these important excretions involves bilirubin. And bilirubin is a waste product from hemoglobin breakdown in the spleen. And it turns out hepatocytes are going to secrete bilirubin into bile, and the normal flora of the large intestine is then going to turn it into urobilinogen, and urobilinogen is going to be converted by bacteria into something called stercobilin, which is actually brown and is responsible for most of the brown color of fecal material. Now, a small portion of urobilinogen is resorbed by the large intestine and ends up back in the hepatic portal system, but the majority of it is just going to be resecreted into the bile. Any of it that is reabsorbed is going to be just resecreted into bile. And then it is important to note that a small portion of this is going to remain in the blood and eventually just be filtered out by the kidneys. And this actually is what gives urine its yellow color normally, because urobilinogen is actually primarily yellow. And when it's finally filtered out by kidneys, that's where the color of the urine comes from. So the same thing gives fecal material its brown color and urine its yellow color. Just interesting. Now, what chemical is responsible for the brown fecal material color? I said a bunch of these, but which one is it that actually is responsible for the brown color? It's stercobilin, the urobilinogen derivative made by those large intestine flora. Remember, urobilinogen gets converted into stercobilin by all those bacteria that live in the large intestine. So that's where the brown color comes from. Now, it turns out that manufacturers of hokum are really good at IDing things that almost sound scientific enough to get you to buy them. And one of these is going to be telling us that Our bodies are toxic wastelands, and they're filled with toxins, and the only thing that can save us, for a price, of course, is their detoxification product. Herbalife, do the wraps. It works. This is a big business, accounting for multi-billion-dollar alternative health industry. And the question that people have is, does it work? But a better question is really, do we need it in the first place? Are our bodies swimming with toxins? Do we all need to detox? No. If you have a liver, it's perfectly good at doing everything that all these It Works, Herbalife, colloidal silver crap says that they're doing. The liver is constantly filtering your blood, and it is a magnificent organ. It works tirelessly to convert any toxins or harmful chemicals into non-harmful substances that are just excreted. And there is no herbal supplement, foot pad, magic bracelet that even comes close to doing what our liver does every day. And what's more important is your liver does it for free. So do not go wasting your money unless that wasting your money is what you feel like doing. Now, as I said, the gallbladder has a very close relationship to the liver, and that it sits on the back of the liver. And what the gallbladder really does is it's going to receive most of the bile from the common hepatic duct. And what it does really is store it, concentrate it, and release it when needed. And this is going to be stimulated by that CCK hormone, which will trigger a smooth muscle contraction in the wall of the gallbladder. And that gallbladder is just going to squeeze the bile into what is called the cystic duct. And the cystic duct is going to join the common hepatic duct to form what is called the common bile duct. Common bile duct is going to join the main pancreatic duct to form the hepatopancreatic ampulla, and this hepatopancreatic ampulla is going to be surrounded by a smooth muscle ring called the hepatopancreatic sphincter, which controls that emptying of bile and pancreatic fluid into the duodenum. Do remember that the contents of this ampulla then empty into the duodenum at a site called the major duodenal papilla. And now bile is going to contain a lot of calcium salts and cholesterol, and both of them can actually solidify into hard lumps called gallstones. And these gallstones are generally going to be asymptomatic, but if they do get lodged in the cystic duct or in the common bile duct, they block the outflow of bile. This can result in tremendous abdominal pain, especially when someone eats a really fatty meal. All those lipids are going to really trigger the constriction of the gallbladder, and if it can't squeeze the bile out, it's really painful. Another symptom that you'll find here is the presence of undigested fat in the feces, and it may also take on a clay color due to the lack of bilirubin excretion. And painful gallstones are really generally treated by a cholecystectomy, the removal of the gallbladder. And it turns out that over a short period of time, the absence of a gallbladder will reduce the ability of the small intestine to digest fats. But over time, it turns out that the common hepatic duct can actually just enlarge and then take over many of the gallbladder's functions. And here you see some of those structures I was referring to. You see the cystic duct here joining up with the common bile duct, and then eventually they become the hepatopancreatic ampulla, emptying through the major duodenal papilla, but not before you see this smooth muscle ring here, the hepatopancreatic sphincter. All these structures are very important. I just want to make sure that all of them are properly labeled. Here you can see an x-ray of the gallbladder with some gallstones in it. And again, the treatment is typically just a cholecystectomy, the surgical removal of the gallbladder, typically an outpatient surgery. Just avoid fatty meals for a while, and there's no really long-term adverse effects. Now, the liver producing bile is generally a continuous rate, but not at a basal rate, like how pancreatic and gastric juices are. Instead, the bile secretion doesn't really occur until the gallbladder contracts and the sphincter at the hepatopancreatic ampulla relaxes. CCK is the biggest trigger for all of this, and then to a smaller extent, the vagus nerve really influences this as well. There are other hormones that influence it, like secretin, which stimulates bile production, and release from hepatocytes. But remember, the most potent stimulus for this is going to be the release. The most potent stimulus for bile production is bile itself, specifically the bile salts. Do remember that bile salts are going to be resorbed by the last section of the ileum and then transported back to the liver, right? And as bile salts re-enter the liver, bile secretion is going to rise dramatically, and it continues. This bile secretion is going to continue into the duodenum until the duodenum empties, at which point CCK and secretin levels are going to drop. And now we should have a figure here. We should have several figures. The first showing the secretion of bile, and then the next showing a summary of the structure and functions of the accessory organs. So you see, first you have the duodenal cells releasing CCK and secretin. Both of them travel through the blood to the liver. CCK is actually going to travel to the gallbladder, contracting those smooth muscles, excreting bile. Those bile salts are going to, and as well as the secretin, are then going to trigger the release of more bile. And so this is just going to be a continuous sort of feedback, a positive feedback loop. Now, what is the biggest trigger for the contraction of the gallbladder to release bile? What triggers the contraction of the gallbladder to release bile? CCK. Remember, CCK triggers that release. Now you see the summary here of these accessory organs: the teeth, tongue, salivary glands, the pancreas, liver, and gallbladder. And I've already gone over these, but do be sure you pause, review if you need to. Moving on. Now we're going to talk about really just an overview here of digestion and absorption. We've talked about the organs and how they work, but now we're going to talk especially about nutrient absorption and those key digestive processes like digestion and absorption. So we're going to look at the physical and chemical breakdown and the absorption of dietary carbs, proteins, lipids, and nucleic acids. And if you remember, the process of digestion is basically just breaking food down into smaller and smaller pieces until only the nutrient molecules remain. And remember, there are two types: mechanical, which involves physically breaking the food into smaller pieces, and chemical, which breaks the chemical bonds. And it turns out all nutrients go through the same mechanisms: mastication in the mouth, churning in the stomach, segmentation in the small intestine. But chemical digestion varies for each nutrient because they have specific enzymes for specific bonds. And most of these enzymes catalyze what are called hydrolysis reactions that use water to break these chemical bonds. An example we should see. Okay, well, so these enzymes catalyze hydrolysis reactions using water, and that does explain why there is so much water secreted with fluids like gastric and pancreatic juices. The more water there is, the more hydrolysis that can happen. And turns out that without water, or without digestive enzymes rather, these hydrolysis reactions would occur, but they would be much too slow for us to actually extract nutrients from the food we eat in a timely manner. And so that's why these enzymes really catalyze the reactions. They speed it up in a process known as enzymatic hydrolysis. And so here you can see one of those amino acid breakdowns in the presence of water. You see a dipeptide, which is just two proteins, two amino acids chained together. In the presence of water, they'll break down, and you'll see two hydrogen atoms and one oxygen atom from the water molecule separating them. Now, as it turns out, once nutrients are broken down to their monomers, they can enter the body. And do remember that the mucosa of the alimentary canal is actually what is called an external membrane. And so remember, even though food doesn't seem like it's on the outside, it technically is on the outside until it's absorbed. And nutrients and other substances must be absorbed across that epithelium of the alimentary canal to enter the bloodstream before they can be delivered to body cells. And as with digestion, this process is going to differ for each nutrient. Now, the breaking down of macromolecules with the addition of water, what is that? I'll give you a hint, it's in the name. Hydrolysis. Hydro means water, lysis means breakdown. The breakdown in the presence of water is hydrolysis. Now, the typical path for a nutrient is going to be traveling from the lumen to the blood, right? And it has to cross barriers. And you can see here the path of a typical substance traveling through an enterocyte of the small intestine. Do note that sometimes small molecules like water can just cross between them instead of through them, but most substances we discuss have to take this route. And the first step is the most significant barrier, of course, is going to be that apical side of the plasma membrane. And this is because most of the nutrients, most substances are going to require some sort of protein transport or a channel to cross the membrane. And if no protein exists, the substance will remain in the lumen and eventually be excreted in the feces. So if they are able to instead travel through that apical membrane, that apical point of the membrane, they're going to travel down through the cytosol and they're going to exit. They're going to again be transported by another protein and transported out the basal side of the plasma membrane into the interstitial fluid, and then eventually from the interstitial fluid into an intestinal capillary entering the bloodstream. But again, remember that apical membrane is really going to be the front line, because if there is no receptor for this carbohydrate, this protein, this lipid or fat, then it's just going to stay in the lumen. An example here could be elemental mercury. Remember those liquid mercury thermometers? In this form, mercury has a really hard time crossing a plasma membrane via diffusion, and there's no transporter protein or channel protein that exists for it. So if someone accidentally swallows some elemental mercury, most of it's actually just going to pass straight through the alimentary canal, completely unabsorbed. Now, if we start talking about carb digestion, it's going to start where? It's going to start in the mouth with all that amylase in the saliva. And so this is going to catalyze those reactions that break down those long sugars into much shorter oligosaccharides. So polysaccharides get broken down into oligosaccharides and polysaccharides, oligosaccharides. And this amylase is typically going to accomplish very little chemical digestion simply because the food is not in the mouth long enough for it to have any effect. Although some minor digestion actually continues in the stomach, but the bigger thing there is the stomach acid, because it actually it inactivates the enzyme before this task can mostly be completed. But chemical digestion of carbohydrates will resume in the small intestine, where polysaccharides that may have escaped and oligosaccharides will encounter something called pancreatic amylase. And this enzyme is very similar to salivary amylase, and it catalyzes the reactions that break the remaining polysaccharides into oligosaccharides. And oligosaccharide digestion is really just going to be completed uh, by enzymes that are products of enterocytes. Remember that brush border? These are going to be called brush border enzymes like lactase, maltase, and sucrase. And lactase catalyzes only one reaction, milk sugar lactose into glucose and galactose. Maltase and sucrase actually catalyze reactions that break oligosaccharides into disaccharides and monosaccharides. And so the end result of this activity is going to be the production of monosaccharides, glucose, fructose, and galactose. And then eventually, after all the carbohydrates have been digested into their simple sugars, their monosaccharides, they're ready to be absorbed. Now, both glucose and galactose are transported across the enterocyte's apical membrane by the same mechanism. Do also note that the higher concentration of glucose and galactose in the cytosol is greater than that in the lumen. And this simply means that absorption of glucose and galactose requires these molecules to move against their concentration gradients. Now, which enzyme is it that breaks down lactose? Lactose enzyme is lactase. So like I said, it does require the glucose and galactose to move across their concentration membrane. And if you guys remember, anytime something is pushed across its concentration gradient, energy has to be expended. And so this comes through a secondary mechanism called the sodium glucose cotransporter. This is a secondary active transport system that uses ATP to indirectly move a chemical across the membrane. In this case, a sodium-potassium pump consumes ATP to drive sodium ions out of the enterocyte and into the lumen of the small intestine, creating a concentration gradient that favors the movement of sodium ions back into the enterocyte. And so this is going to allow sodium ions to fuse back into the enterocyte, driving the transport of glucose and galactose as well into the enterocyte. So it creates a sort of a vacuum as these are pushed out, as the sodium is pushed out, a sodium glucose cotransporter is then able to push sodium back in, but it will also carry some glucose or galactose. And it turns out fructose is really unable to bind to the sodium glucose cotransporter, so it actually uses a separate mechanism. Fructose will instead bind a channel that mediates its facilitated diffusion across the membrane. And so facilitated diffusion is really a passive process, and so it doesn't really require any ATP. And a concentration gradient has to be present, so there is going to typically be more fructose in the lumen than there is in the enterocyte. But all three monosaccharides tend to cross the basal enterocyte membrane with the same facilitated diffusion mechanism, involving a membrane protein very similar to the one that helps fructose cross. And after these monosaccharides manage to cross the basilar membrane into that extracellular fluid, they will then go into the capillaries in the villus, and then once they're in the blood, they're delivered to the liver via the hepatic portal vein for their processing. So we see here the various monosaccharides being diffused across the membranes. First, the polysaccharides need to be broken into oligo and disaccharides. Then the brush border enzyme is going to catalyze the breakdown of disaccharides into simple sugars. So now we're going to have glucose, galactose, or fructose. Excuse me. So glucose and galactose actually have to bind to that sodium glucose cotransporter at the same time that sodium potassium pump is going to it's going to pump sodium out of the cell so that sodium up here, let's say it's going to pump sodium out of the cell down here so that this cotransporter up here can pump sodium in, but it will also manage to bring in this glucose or galactose. That gradient is going to drive, the transport. And fructose is a little bit different. It just gradually binds to a protein here and diffuses in. It's just facilitated diffusion. As long as there's more fructose up here than there is in here, everything's good. Fructose will diffuse across the membrane. And then once all these monosaccharides, all these simple sugars are in the enterocyte, they all do the same thing, which is just facilitated diffusion across the membrane via a transporter protein. Once they're out, they're going to diffuse into the blood after they diffuse into the interstitial fluid, and then they're going to travel to the liver. So it's a matter of active transport, passive transport, sodium potassium pumps, sodium glucose cotransporter pumps, and enzymatic breakdown. If you guys have any questions about any of this, don't hesitate to contact me. Now, it turns out that many, many people actually have a real issue producing the enzyme lactase. And so they can't digest sugar milk. And I'm one of them. I don't know what happened, but once I hit my late teens, I stopped being able to handle my dairy. And it turns out that about 75% of adults actually show this decreased lactase activity. And it really actually has something to do with ethnicity. About 5% of Northern Europeans are lactose intolerant, but about 90% of certain regions of Asia are actually lactose intolerant. And so without the body being able to produce lactase, lactose can't be digested, right? And so this undigested lactose remains in the small intestine until it's delivered to the large intestine. And when it gets there, oh boy, lactose in the large intestine really acts as an osmotic particle that can draw lots of water into the feces. And so this leads to the characteristic abdominal cramping and diarrhea when lactose is consumed. People generally are able to manage their lactose intolerance by either taking over-the-counter medications or avoiding lactose-containing foods. Those medications typically actually contain a synthetic form of lactase. It's just a supplemental enzyme that you're taking. I know I take those Lactaid tablets. I just chew them up. And it works pretty well. Now, we've talked a lot about carbohydrate digestion and salivary amylase and lactase and all these enzymes. But what about proteins? Well, we've already kind of talked about this. And it turns out protein digestion doesn't actually start until the food content reaches the stomach. And if you guys remember, chief cells inside the stomach glands, those gastric glands, they're going to produce pepsinogen. And when pepsinogen hits that pH of about 2.0, it becomes pepsin. And pepsin is completely inactive at a basic pH, right? A neutral pH, rather, pH of 7. And activated pepsin is going to catalyze reactions that digest proteins into smaller polypeptides, oligopeptides, and some relatively free amino acids. And although the reactions that are catalyzed by pepsin, they're only going to digest about 10 to 15% of the proteins. And individuals who do not produce any pepsin at all actually don't appear to have any less ability to digest these proteins. So pepsin may not actually be tremendously useful. Only 10 to 15% of protein is catalyzed by those reactions. The majority of that protein digestion is going to take place in the small intestine through those brush border enzymes and the pancreatic enzymes. And the 5 pancreatic enzymes that digest proteins are all going to be released as inactive precursors. This is basically going to protect the pancreas from a process called autodigestion. Basically, it means these precursors aren't going to be able to digest the proteins of the pancreas itself. And the first one of these pancreatic enzymes is called trypsinogen. And when it becomes released and active, it becomes trypsin, the enzyme trypsin. And trypsin activates when it encounters several enzymes on the intestinal brush border. Trypsin is going to catalyze reactions that convert other pancreatic enzymes to their active forms. So this is one of those kickstarters where trypsinogen gets activated into trypsin, and then trypsin causes a sort of cascading reaction to activate the other pancreatic enzymes. It also activates additional trypsinogen as it is secreted by the pancreas. And these enzymes are going to catalyze reactions that digest, remember, proteins, polypeptides, into oligopeptides and some free amino acids. So aside from the pancreatic enzymes, you also have brush border enzymes coming from the enterocytes. And these are going to catalyze the digestion of oligopeptides into free amino acids. Now, these enzymes are actually kind of limited in the scope of what they can do. So the reactions that they catalyze typically leave small oligopeptides, like 2 to 3 amino acids. They remain undigested. And so for these small oligopeptides, they get taken into the enterocyte, into the enterocytic cytoplasm, and they are going to encounter more enzymes in the cytoplasm. They're going to further break them down into free amino acids. So to be absorbed, you remember they have to cross that brush border. And so it turns out these proteins consumed for nutrition need to be broken down into the small oligopeptides and free amino acids. But humans do have the ability to absorb small amounts of whole undigested proteins that may be involved in immunity by the process of endocytosis. So specialized cells overlying what are called the Peyer's patches in the small intestine are going to ingest these proteins and deliver them to the lymphatic tissue inside of those patches. This is really, really important for the development of newborn infants' immune systems. And oligopeptides, when we're actually talking about crossing that apical enterocytic membrane, are going to primarily be transported by secondary active transport membrane proteins. This is going to be very similar to what we've already seen. The sodium-potassium pumps activate, and when they're brought in, the oligopeptides are going to be broken down into the free amino acids, which then exit the basal membrane of the enterocyte via facilitated diffusion. After which, they're going to go into the capillaries of the villus, remember? Now, just like carbohydrates, these amino acids are now going to be delivered to the liver for processing via the hepatic portal vein. And here we can see some of this process here. We see a fully formed protein out in the lumen in the small intestine. We see that here. It gets broken down. In this case, we're just going to simplify it and say it's all broken down into free amino acids. The amino acids are going to be attached to a secondary active transport protein, okay? And then once this sodium potassium pump down here pumps enough sodium out, it's going to create that gradient, right? And so this active transport protein here is going to pump sodium in, and as it does that, these amino acids are going to flow in with it. And so once these amino acids enter the enterocyte, right, then they're going to make their way down to the basilar membrane, and they're going to cross it via facilitated diffusion, essentially another high to low gradient concentration. And so they're just going to be transported out of the cell into the interstitial fluid and eventually the bloodstream. And remember, once they're in the bloodstream, they go to the hepatic portal vein in the liver. Now, it turns out pepsin only catalyzes what percent of ingested protein reactions? How much protein is digested by pepsin? It's 10 to 15%. Remember, it's very, very marginal. And so now that we've gotten those out of the way, let's talk about lipids. And when I say lipids, it can include anything. There's a wide variety of oils and fats that are included in this category. But about 90% of dietary lipids are called triglycerides, and that's what we're going to discuss here. If you guys remember, these are going to be 3 fatty acid monomers bound to a 3-carbon core called glycerol. So it's a triglyceride. And if we look at actually the lipid digestion, it's going to be more complex than either the carbohydrate or the protein digestion, simply because lipids are nonpolar. They're hydrophobic. And so it causes them to stick to one another instead of interacting with the water. And so what happens here is these lipids form large clumps in a water-based solution. Think of if you've ever had a sink full of dishes and you put a really greasy dish in there. On top of the surface of water, you're going to see all this fat sort of globbing up together. If you've ever been particularly lazy and left a dish soaking overnight and you come back in the morning, you'll find that ring around the edge of the sink at the top of the water line. Those are going to be mostly lipids. And so that's exactly what's happening in your digestive organs. It's going to float on the top of that chyme. And so these digestive enzymes really have very little surface area if this clumping is allowed to happen inside of the alimentary canal. So the first thing that has to be done is really these lipid globules are going to have to be physically broken. And this is going to give digestive enzymes more surface area on which to work. First thing that's done, mastication. You're breaking down those fat molecules with your mouth. Then it's going to be churned in the stomach and segmented in the small intestine. But even after all these processes, lipid globules can still be too large for chemical digestion. To facilitate this, to really mechanically break lipids into even smaller molecules, we need the bile salts. And the bile salts, if you guys remember, are amphiphilic, which means they have both a polar and a nonpolar part. We should see the structure here of a bile salt. And you see this polar hydrophilic section up here, and then you see this nonpolar hydrophobic section over here. So the nonpolar portion here is going to stick to the lipids, and the hydrophobic portion here is going to freely interact with the water. And so that physically is able to break up lipid globules into smaller pieces. This is a process called emulsification. And emulsification, it results in multiple tiny liquid droplets, which can each be coated with bile salts. This essentially gives digestive enzymes enough surface area to work on, which can easily help digest the lipids. If you think about it just as a better analogy, imagine 1,000 people trying to grind the moon into dust with hammers and chisels, right? They're going to break off pieces with their hammers and chisels, but progress is going to be so slow that it's really non-existent. And so that's what's going to happen with lipase trying to catalyze large unemulsified lipid globules. Now, imagine the moon is split into 7 billion pieces, and 7 billion pieces are people are each given a small piece of the moon, as well as a hammer and a chisel. The entire moon is going to be ground to dust much more quickly and efficiently this way. And so that's what we're talking about when we say emulsification. It's split into multiple tiny pieces so that pancreatic lipase enzymes then have a huge amount of area on which to catalyze their reactions. But the actual process is what? First, the lipids are going to be broken apart by churning and broken down in reactions catalyzed by gastric lipase. Gastric lipase is going to catalyze the reactions that remove at least one fatty acid from triglycerides, leaving some free fatty acids and diglycerides. This converts about 15% of dietary fats. After this, the lipids are going to enter the small intestine and be emulsified by those bile salts. So these are going to these bile salts are going to coat the lipids, physically breaking them apart. And remember, this is not a chemical process. There are no bonds breaking. This is purely mechanical. After these bile salts coat these lipids, pancreatic lipase is going to be released, and it's going to catalyze the lipid breakdown. It's going to break these triglycerides down into monoglycerides and several free fatty acids. Any bile salts that remain are going to be associated with these digested lipids, and they're going to form a structure called a micelle. A micelle is going to essentially be a surrounding around the lipid. It's think of it sort of as like a prototype cell. And so here you can see some of these processes of lipid digestion. In the stomach, you see it's going to be churned and broken apart. But of course, you do see that large lipid globule here that would normally just float on the top of this water. Gastric lipase is going to break it down into diglycerides and free fatty acids. Eventually, it's going to travel into the small intestine and become emulsified by bile salts. So you see these lipids, these smaller lipid globules here get coated by these bile salts, and it produces these structures here. Once those emulsified lipids are produced, these bile salts are all bound to them. They're going to have pancreatic lipase attached to them. You see that here inside the small intestine, remember, and it's going to break them down into free fatty acids and monoglycerides. Now, if there are any bile salts still there, they're going to be associated with these lipids, these digested lipids, in what is called a micelle. And you can see that here. It looks almost like a phospholipid monolayer, right? It looks so like if it was a regular phospholipid dilayer, or it would look like this, with the phosphorus head and the two lipid tails touching each other, providing a hydrophilic head and a hydrophobic tail. Well, we see only one of these here. We see the hydrophilic head here and the hydrophobic tail here attaching to the free fatty acids and the monoglycerides, rather. And so again, it helps to think of a micelle as a sort of prototypical cell. Like it almost looks like a cell membrane. And this sort of just helps to push these through so that they don't get stuck. Now, for the absorption of lipids, it's going to be just as complicated as the digestion. Again, because these lipids are nonpolar, they're going to face several water-based barriers that are going to prevent them from passing into the cytosol. Like the mucus lining of the small intestine, the polar phosphate heads of the plasma membranes. And so lipids really require some assistance to approach the enterocyte and eventually cross them. First, those micelles that we've talked about, they're going to work to escort the lipids to the plasma membrane. Remember that these bile salts have their polar portions on the outside and their non-polar portions on the inside. The outer polar portion allows this lipid to get close to the enterocyte and approach that polar mucous membrane. These lipids are then going to be attracted to the fatty acid tails of the phospholipid bilayer. So this is just going to be simple diffusion where once they get close, they're able to diffuse across a membrane. Typically, some lipids may use facilitated diffusion, but most of this is just simple diffusion. And then any leftover bile salts from the micelles are going to remain in the lumen of the small intestine and be reabsorbed in the large intestine. And then once the lipids are inside the cell, the enterocyte, they're going to be reassembled into triglycerides and packaged into structures packaged with cholesterol, several other lipids, phospholipids, and lipid-binding proteins called apoproteins. These are going to be bound up in structures called chylomicrons. And chylomicrons are similar to these micelles in that they have a nonpolar face and a polar portion on the outside, a nonpolar facing inside and a polar portion facing outside. And this is going to be what allows them to travel through the blood, through that polar water molecules in the blood. So the chylomicrons are going to be released out the basilar membrane via exocytosis of the Golgi. The Golgi apparatus in the base of the cell is going to package these up, and then it's going to exocytize out into the interstitial fluid and eventually into the bloodstream. Actually, I'm sorry, it's not going to go into the bloodstream. It's going to these chylomicrons are too large to enter the capillaries. And so what they actually do is they go into the lacteals. If you guys remember, the lacteals are going to be the lymph portions of these villus, the central core of the villus. We should see here in just a little bit. Now, unlike proteins and carbs, lipids aren't taken through the hepatic portal vein. Not directly, anyway. Instead, the hepatic portal vein is going to deliver any leftover bile salts back to the liver, where they can then make new bile. But the lipids are going to travel within the chylomicrons through the lymphatic vessels and eventually to the thoracic duct. And this is going to be where they join the blood with the rest of the lymph as the thoracic duct meets that left internal jugular and left subclavian veins. Now, as the chylomicrons travel through blood capillaries, lipids are progressively removed and enter cells. So it's sort of a gradual removal of chylomicrons, of lipids from the chylomicrons. Eventually, the liver does receive them, and the lipids are going to be removed from the chylomicrons in the liver. These are then going to be processed by a number of metabolic a number of metabolic processes. So once the chylomicrons have been emptied, they're taken in by these hepatocytes and they're just taken apart. And so here you can see that basic transport that we were talking about in the small intestine. You'll see the micelles are approaching. Up here, you see a micelle approaching with its polar face and its non-polar tail on the inside. So they're going to get, these micelles are going to escort the lipids close to that plasma membrane. And then once they get there, the lipids are going to be able to diffuse across that phospholipid bilayer. Remember, they love non-polar things, and the phospholipid tails, or the phospholipid bilayer, has those lipid tails that they can just straight diffuse across. Now, once they're inside, they're going to reassemble into triglycerides, and then the Golgi apparatus is going to package them into chylomicrons. And then once they're packaged, the Golgi apparatus then is going to release them via exocytosis. And remember, they don't go through the blood, they go through the lymph in the lacteal. So that shows that process. You see triglycerides being reassembled here. Oh, I'm sorry, here. Couldn't find where my mouse was. And then you see this Golgi apparatus producing this exocytosis package here, where it can then just be pushed out. And this is just going to be that summary of how each of these macromolecules are digested. We've gone over this a little bit, so if you want to go ahead and just sit here and review, just pause this and make sure you understand which enzymes coming from where assist in what manner of the digestion. You're more than welcome to do that. But moving on. Now we have another macromolecule. Remember, it's not just proteins, lipids, and carbs. We also have nucleic acids. And these acids, these nucleic acids, are going to begin chemical digestion in the small intestine via pancreatic nucleases. These nucleases break nucleic acids down into their individual nucleotides. Eventually, this digestion is going to become more advanced as you get to the brush border. The brush border is actually going to remove phosphate groups and sugars from nucleotides to leave naked phosphates, riboses, or deoxyriboses, as well as a nitrogenous base. Once all those are created, they're then going to be absorbed via primary and secondary active transport mechanisms into capillaries in the villus. So after that, the hepatic portal system is going to take them in via the blood, and they're just going to take them to the liver to be metabolized. So this one's really relatively simple. Nucleases break the nucleic acids down to a certain degree, and then once they get to the brush border, they're going to be attacked by several more enzymes that leave naked phosphate ions, riboses, or deoxyriboses. Essentially, it's going to be a phosphate, a sugar, and a nitrogenous base. And then those substances are going to be transported across the capillaries into the villus. Now, what pancreatic enzyme, remember this is a pancreatic enzyme, what begins digesting nucleic acids of food? It's a pancreatic enzyme that digests nucleic acids. I tried to trick you by putting extra emphasis on pancreatic. It doesn't matter. It's still nuclease. So here we've got the list of several of these enzymes, their sources, and the reactions they catalyze. I have talked about all of these, so please feel free to just pause, review as you need. But I'm going to continue on. So the last thing we need to talk about as far as digestion and absorption goes is water, electrolytes, and vitamins. So now the majority of what we're talking about occurs in the large intestine. A significant quantity of occurs in the large intestine. Of course, the majority of it still occurs in the small, but let's look at the absorption of water. So typically, about 9 liters of water are going to be dumped into the small intestine every day. 2 liters of it are going to be ingested, and the remaining liters are secreted into the alimentary canal on its own. It's going to be secreted by the alimentary canal and accessory organs. Now, out of these 9 liters, about 8 of them are absorbed by the enterocytes of the small intestine. The remaining water is going to be absorbed into the enterocytes of the large intestine, leaving about 1/10 of a liter to be excreted in feces. Now, it turns out water absorption occurs only by osmosis, so there is no active transport here. And this requires the cytosol and the extracellular fluid of the villi and the enterocytes to be more concentrated than the fluid in the lumen of the intestine. For this reason, anything that causes excess solutes to remain in the lumen, that can cause water to be held in the lumen and decrease its resorption. For this reason, osmotic laxatives these are called stool softeners, and these drugs are going to consist of solutes that are either not absorbed or given in amounts of excess that can be absorbed. So essentially, they're able to withhold water from the enterocytes of the large intestine, making the stool more watery, making them softer and easier to eliminate. Now, electrolytes like sodium and potassium, they're going to be taken in by the diet, and they're going to be present in the secretions from the digestive organs. Most of the time, these are absorbed by the small intestine, but significant amounts do absorb by the large intestine. You've already seen how monosaccharides and amino acids co-transport sodium, and this sodium absorption is key in the absorption of anions. So as a sodium ion enters the enterocyte, this creates an electrical gradient that's going to drive the co-absorption of anions, negative ions like chloride and bicarbonate. And so several other electrolytes that we've talked about have specialized mechanisms. Calcium is only absorbed with the help of vitamin D. And then certain electrolytes that are present in smaller amounts, like iron and magnesium, require active transport to be absorbed. Now, what exactly are vitamins? You guys have heard about vitamins, but these are chemicals that are normally provided by the diet that could be involved in a host of metabolic reactions. There are 2 types of vitamins. There are water-soluble vitamins that are polar. Remember, water-soluble, so it has to interact with the water. And then there are fat-soluble vitamins that are lipid and non-polar based. Most of the water-soluble vitamins are going to be absorbed by the small intestine, simply diffusing across the enterocyte's membranes. There is an important exception called vitamin B12. Vitamin B12 has to bind to intrinsic factor, which is produced by, again, remember, those parietal cells of the stomach. After B12 is bound to the intrinsic factor, it can be absorbed by the ileum of the large intestine. No, I'm sorry, the ileum of the small intestine. Sorry, I was confusing ileum and cecum. Any shortage of intrinsic factor can actually impair B12 absorption, and we'll talk about that in just a second. But as far as fat-soluble vitamins like A, D, E, and K, those are going to be covered a little bit more in the metabolism and nutrition chapter. But I just want you guys to remember, fat-soluble vitamins include A, D, E, and K. Now, which of these is not a fat-soluble vitamin? No, I just told you all of them, right? All four. Vitamin C was not one of them. A, D, E, and K are fat-soluble vitamins. So vitamin B12 is that crucial vitamin for several metabolic processes, like the formation of red blood cells, normal functioning of the nervous system, and it's commonly associated with quote-unquote energy. Typically, that's just the normal functioning of the nervous system that it's assisting. Now, if intrinsic factor is going to be absorbed, okay, or I'm sorry, if B12 is going to be absorbed, it requires intrinsic factor. And so it creates what is called an IF-B12 complex. This IF-B12 complex is going to bind to the enterocytic plasma membrane, okay, and then it's going to be able to be absorbed across the membrane. And there are several conditions that can impair the production of intrinsic factor. Stomach autoimmune conditions, atrophy of the gastric mucosa, surgical removal of the stomach for weight loss or cancer treatment. All of these can impair how much intrinsic factor is produced. So without it, you become B12 deficient, even if there's enough B12 in the diet. This B12 deficiency can lead to a condition called pernicious anemia and nervous system problems that can really only be treated with intravenous B12 injections. And so here you can see some of the big picture of digestion. You see as food enters and is ingested by the mouth, teeth and tongue masticate it, they chew it up, and then they begin to deglute it, they swallow it. And this swallowing reflex is going to propel the bolus to the stomach, as seen in that second figure there. Let me get there. So deglutition pushes food into the pharynx, down the esophagus, and now it is in a form called a bolus, right? Well, that bolus is going to be churned and chemically digested in the stomach and turned into a liquid now called chyme. Once the chyme is secreted out the stomach into the small intestine, it's going to be bathed in pancreatic juice and bile. And when that is happening, the nutrients, this chemical digestion is going to be occurring, and nutrients are going to be passed through the enterocyte cell membranes into the blood and the lymph tissue. Eventually, that material is going to pass into the large intestine, where water, electrolytes, and vitamins are either going to be reclaimed, like resorbed, or they're going to be just claimed out of the remaining chyme, like vitamins A, D, E, and K. Eventually, the unused, undigested material is going to be excreted in feces through that process we covered called defecation. And that is digestion. That's all of it. So, if you guys at any point have any questions, we do have a couple of just test yourself questions that I'll go over really quick. If you guys remember, what do goblet cells secrete? Goblet cells secrete mucus. Don't forget that. And then the inferior most portions of the pharynx is called, what's the lowest portion of the pharynx? It's the laryngopharynx. Remember, oro, mouth, naso, nose, laryngo, throat. So the throat is going to be the lowest. And then which cells of the stomach produce and secrete hydrochloric acid? Which ones produce hydrochloric acid? It's the parietal cells. Those are the ones that produce this all-important stomach acid. All right, that's it. That's all we have for this week, guys. Please review. Contact me if you have any questions at all. I will see you in the next one.